Okay. Good morning, afternoon, evening, everyone, and welcome to the Palaeoverse Lecture Series. This lecture series is focused on software development and methodological approaches in paleontology with the aim of promoting the use of new tools in the field and to give researchers a platform to showcase their work. With this series, we hope to shake up the usual type of lecture by having a mixed format with time dedicated to the presentation, demonstration, and discussion of the software or method presented. Typically, these sessions will run for about 50 minutes in total, and we will run this series throughout the year as we have, generally on the last Thursday of each month, unless they overlap with holidays like today, or this month. If you're interested in presenting a new method or software you've been developing or implementing, please feel free to reach out. We are always happy to hear from the community. So before moving on, just a few quick introductions. My name is Will Gertie, and with me today I have Aaron Dillon and Lewis Jones. The three of us are on the core team of Palaeoverse, an organization aiming to bring the paleobiological community together to share resources, reach agreed standards, and improve reproducibility in paleobiological research. If you want to know more about Palaeoverse, please check out our website and GitHub organization that are being put in the chat now. And that's all from me for now. Aaron will now give you some helpful tips and instructions for this webinar, and Lewis will introduce this month's speaker after that. Thank you so much for being here, and please enjoy the lecture. Over to you, Aaron.
Wonderful. So as Will mentioned, the format of this seminar will consist of a talk by our speaker, followed by about 10 to 15 minutes at the end dedicated to Q&A and discussion. My fellow hosts and I will help field your questions and moderate this discussion. Please note that the seminar will be recorded and that it will be made available on the Palaeoverse YouTube channel. However, we will stop recording before the Q&A period in case you prefer not to be recorded. So before we get started, I'd like to quickly point out a couple of useful Zoom controls at the bottom of your screen. So you'll find a Q&A button. You can use this to submit your questions to our speaker, and then during the Q&A session, we'll either read out your question or invite you to turn on your microphone. There's also a raise hand button. So alternatively, you can raise your hand and we will call on you and invite you to turn on your microphone so you can ask your question. There's a chat function. So currently you can only chat with the hosts in case of technical difficulties, but we'll allow you to chat with the other attendees during the Q&A period. And then there's a closed caption button that you can turn on and view the transcript at any time. So lastly, I'd like to highlight a couple of ground rules. Our aim is to cultivate an open, respectful, and inclusive environment during this lecture series where we can all learn from each other. So with this in mind, we ask that you please be respectful and kind when engaging with the speaker and fellow attendees, particularly as you ask questions and interact with others in the chat. Any form of harassment or inappropriate behavior will be grounds for removal from the webinar platform. For more information, please refer to the Palaeoverse Code of Conduct, which can be found on our website and I believe is now in the chat. And with that, I will pass it over to Lewis to introduce today's speaker.
Thanks, Will and Aaron, for getting us all up to speed on how the lecture series will work today. So I've got the great honor of introducing our speaker, Dr. Juan Cantalapiedra. Juan is a paleobiologist and senior scientist at the National Museum of Natural Sciences in Madrid, Spain. Now, Juan's research mainly focuses on mammalian paleobiology and paleoecology, usually applying phylogenetic methods, but also through the analysis of large fossil data sets. He is particularly interested in the multiple factors that shape diversification rates through time, such as phenotypic innovations and competition. Increasingly, Juan is using Bayesian approaches in his research to combine morphological and genomic data to estimate evolutionary trees. Now, aside from his research, it's fair to say that Juan is also a great educator and has been offering several courses through transmitting science on macroevolutionary analyses for quite some time now. So without any further ado, we're really excited to hear from Juan today with a talk titled Mammals as a Key to Evolutionary Theory. Now, please join me in welcoming Dr. Juan Cantalapiedra to our virtual stage. Over to you, Juan.
Thank you. Thank you so much, Luis, and thank you all the core team in Palaeoverse for inviting me. It's my pleasure to be here. Today I will be talking about macroevolution in mammals. And the title, when you guys asked me for a title, I was like, well, I will give them a title that is general enough for me to then think about things I can squeeze in. And I borrowed this title from this paper by Elizabeth Berba. I think this is 1992, 1993, I think. And this is, a very key figure in paleobiology, in modern development of paleobiology. She passed away last February, so I wanted to take the chance to talk a little bit about her. And she was very good at, thinking about processes that would propagate what happens at the organism scale to macroevolution. And I think this is one of the critical frontiers that in our discipline today. And of course, methods have improved a lot. So we have a good opportunity to bring questions and theories developed by people like Elizabeth Vega to new groups, to the same data sets, to improve data sets, etc., etc. So I will be talking today, trying to bring a little bit of this philosophy of macroevolutionary thinking to talk about mega herbivores. I will talk about two different studies we've been working on. One of them is published, the other one is in review right now. And this is because apart from being very conspicuous and nice, beautiful species, these guys are very important for different ecological roles. And of course, there are two main things that we don't know much still about the processes behind, and these are how these things emerge. So how you start having the first mega herbivores, things that are close or above 1 ton in weight, and how they collapse. Of course, when we talk about the collapse of the largest herbivores, we always think about the two processes, either human activities or climate change, and how this can impact, or if there's a combination of the two. So these two questions, their emergence and the collapse of the largest herbivores, is what I will be talking today. For the first thing, I will be talking about evolutionary trends within a given clade. So this is one aspect that I like usually to tackle in my research, to just focus on one clade and just try to understand what's going on in that clade. The other is to look at a broader taxonomical scale, looking at the evolution of fauna, so continental faunas or a regional fauna or something like this. So that will be the second part of my talk, and this is a paper that is now in review, so it's still, we could say, in development. So the question about how mega herbivores, mammalian mega herbivores, came to emergence is very interesting, because we know after the mass extinction of the Cretaceous Paleogene boundary, mammals that survived were small, but then they very rapidly went up to weight hundreds of kilos, and many of them above the ton. And here in this phenogram in orange here, these are brontotheres. Brontotheres, you can see these are the first mammalian group that really explored the mega herbivore needs. Most of the species, or more than half of the species of brontotheres, they weighed above 1 ton. So we wanted to know what was going on with this group, which is a group within perissodactyls. They are related to horses and tapirs and rhinos today. And we put together a lot of information for this paper that was led by Oscar San Isidro, and also had in it Matthew Milbuckler, a brontothere expert from North America, and Oscar is an expert on rhinos and other groups of perissodactyls. And it's cool because, brontotheres, they were at the core of evolutionary discussion in the 20s, and many times they were used to defend views of evolution that were not really very Darwinian. So, just these progressive lines of evolution with mechanisms of recapitulation and things that went not very in accordance to Darwinian thinking. Many of these ideas, including some more updated Darwinian thinking, were put together in what's called generally the Cope's rule. So this is a little bit of a mix of different ideas coming from different evolutionary notions. And we wanted to revisit this idea of what's the mechanism that propelled this clade to really large sizes. So the starting point would be like this very simple model where everything gets really big. Then we wanted to explore this kind of reformulation that Simpson proposed in the 40s, where we really are picking up this part of a signal of a successive radiations or several events of radiations, and each of them are related to different sizes or slightly related to different sizes. And finally, the latest formulation by Stanley in the 70s, picking up this idea of punctuated equilibrium, where we would not expect to see a lot of change within the lineage within each species, but differential survival of each lineage would just make up this trend. So if we have some differential speciation and extinction, we expect to have this trend piling up over time. So we're looking in this last case to not just looking at how the evolution of body size operated across the phylogeny of brontosaurs, but also we really needed this other aspect, which is differential speciation or extinction or both of brontosaurs related to body size. So because Luis told me to talk about methods a lot, here we go. This is a pipeline that Oscar put together for the paper. So we start with these measurements, which is like the standard in mammalian paleobiology to estimate body sizes if you don't have access to a lot of postcranials. So we did data imputation, and we came up with body mass estimates for around 60 species. Then we use tip dating approaches, combining occurrences and morphological matrix in BEAST2. And then we came up with a distribution of millions of trees, and we retained 1,000 trees for further analysis. And on these trees, we run different models of evolution. I will talk about them a little bit in a second. And these models were run into packages. An R package called Geiger and FossilBM, which is a hybrid implementation in Pirate, and I think it's Python or C+ or something like this, and you can run this in different software, and it gives you some interesting shifting trends and shifting rates across the tree. So it operates really well for trees, even for large trees. So it's a very interesting package. And the idea was to just, one of the beautiful things I like about phylogenetic models is that you can bring these three major scenarios of evolution and just make models that resemble that evolution, and then compare these models to the data. And as you can see there in the top, based on maximum likelihood fit, the model that best explained the evolutionary history of body size in brontosaurs was one where time didn't matter that much. It was just how many speciation events separated the species, what is called the kappa model in Geiger. Basically explains the diversity of body size better than time itself. So this is the signal that you would expect if most of body size evolution happened at the speciation events, which is one of the predictions of the punctuated equilibrium. And of course, this model has no direction. So we will talk about how you get a trend without evolutionary direction later on. So this is the main figure of the paper. You have a phenogram at the top, and then in the bottom, we have this figure you can produce with FossilBM, in this case, showing shifts in evolutionary rate in body size across different lineages. We observe, interestingly, that major shifts occur going from very big sizes to very small forms. So apparently, there is more capacity in this clade to evolve into small things really, really fast in time. So the fastest rates are associated to reductions in body size. And one thing that we wanted to be sure about is, we have these phylogenetic models, but of course, this is paleobiological data. How sampling would affect our ability to recover the proper model? So is this speciational model something that comes because it's an artifact of not having a clay that is not very well sampled, which is probably not the case because this is a clay that is very well sampled. But just in case, and we ran some simulations, and this is buried very deep in the supplementary, but I invite you to go through it if you are interested. And is that the take-home message here is that even if you simulate a model of a speciational evolution, so you know the evolution of this simulation is concentrated in the speciation events, the moment you start to degrade the tree and you are less able to sample a species, this model is more and more recovered as a gradualistic model. So this means that even if something evolved in a speciational fashion or in a fashion coherent with punctuated equilibrium, if you don't sample that phylogeny enough, you can still, your test will give you the wrong answer, telling you the evolution of whatever trait was gradual, even though it was speciational to begin with. And of course, you can play with different strength of the kappa parameter, which is the top plot, or the number of tips of the phylogeny. But you can see that when you degrade the tree below 50% of the original diversity of the tree, you start wrongly recovering a gradual model instead of a speciational model. So that's something that was interesting to find. Okay, so as I said, because we have this non-directional model, the kappa model has no direction, so how you get basically a clay that goes into bigger and bigger and bigger, overall bigger and bigger and bigger sizes, we need to call mechanisms like the species sorting or species selection. And we use Pirate to test for this. And Oscar did a terrific work putting together a huge database, because the scenarios we were thinking about is, okay, how body size can affect survival or speciation. And we thought about two possibilities. One is like modulating the predation pressure, because size is important when regulating the pressure depending on the size of the predators that are present in your community, or the overlap, the ecological overlap with other herbivores. So this niche saturation that we have there in green. So Oscar did a huge effort at reviewing body sizes of species that lived in sympatry with each Brontotheriid species, and he tailored down an index for each Brontotheriid species based on the presence in sympatry of herbivores and carnivores, and based on the body size of these herbivores and carnivores. So we were able to feed these shifting niche overlap and predation pressure for each species of Brontotheriid over time, and feed this to a Pirate analysis of, so these things would be regulating speciation and extinction. And we discovered that niche saturation was the best predictor for the patterns of speciation and extinction. And here you can see in this plot is basically a plot where you have a line which is the baseline speciation and extinction for the group, and then each point is a species based on how saturated was its herbivore niche according to body size. So that, interestingly, things that were smaller would be under larger competition, because there are more herbivores in the same size category. So they would show higher turnover in general. So it's not just higher speciation and lower, higher extinction and lower speciation. Both were higher in the smaller things, but the speciation was disproportionately higher. Extinction, was disproportionately higher. So the net balance here was that larger things would just generally last longer, and they were able to produce more species. So these things combined is what drove the trend that we saw. So it's just, we didn't necessarily see changes within the species, but we have this process of species sorting where you have smaller things just going extinct super fast, and this will trim the tree, so that the only way the tree has to evolve is through larger and larger body sizes. So this is, as I said, one example of a study that we do when we focus on one clade. And I think looking at these two facets of cladal evolution is going to be more and more important in the future. Most of the time we see studies that focus only on the phylogenetic part of how the trait evolved from the phylogeny, but we see more and more now with these methods like Pirate that you need to account for really this differential species proliferation to really look at the whole picture. And this is interesting because it's a way to disconnect microevolution from macroevolution. So you can still have a species emerging, so they are still created by local selection, but their fate, how they will survive, is going to come from maybe new settings or environmental forcing that was not, of course, anticipated or things like this. So this would be the first block of the talk. And now I will talk a little bit about a project that we've been working for many years because we are, this is a very, very complex approach to faunal evolution. And I will be talking about the decline of megaherbivores. And I like to start this always talking about when we go today to African savannas and we see all these species of rhinos, elephants, and big antelopes, or etc., etc. We think that this thing has been there for forever. And the truth is that not so long ago, our ancestors were living with a much larger and diverse community of larger herbivores. And this is a beautiful picture that Julius Sotoni put for a press release a few years ago for another paper, where we have Turkana around 3 million years ago, and you have this Australopithecus in the middle of this beautiful party of huge things. So we wanted to really look at the decline of mega herbivores in Africa and try to grasp its timing and the factors that drove this decline. This is a very choral work from with people from many different institutions, curating data, developing software. So there's been a huge, amazing collaboration. So back to mega herbivores, one of the interesting things that now has sparked more research around these things is that we know that when they disappear, it's not just we lose some charismatic, amazingly big species, is that when these things disappear, ecosystems lose a disproportionate amount of ecological roles. So this is a very interesting theory of the loss of, ecological space in Africa as many of these very large things disappear, and how even though there are not so many species, the ecological space really shrinks to less than half. And the interesting thing is that starting in 2018, this paper by Tyler Faith and collaborators showed that the decline of megaherbivores in Africa was not just associated with, humans overkilling things in the last 70,000 years or 100,000 years, but local, and each point in the figure on the left is a locality, so local relative diversity of megaherbivores compared to actual faunas has been decreasing for 4 million years. So this is a really long process. And in a paper Faisal Bibi and I put together a couple of years ago, we wanted to look at this process, but instead of at the species level, at the individual levels, looking at abundance, relative abundance, and relative biomass of herbivore individuals in different localities. So each point there is a locality, but we are not looking at this is a taxonomy-free approach, and we saw that in the larger size categories, so basically again everything above 1 ton, we see this decline both in abundance and in many cases also in relative biomass. And of course, both of these scenarios, this protracted decline has been linked to changes, essential changes in CO2 atmospheric concentrations, changes in the vegetation cover and habitat heterogeneity, and how vegetation essentially changed, changes in productivity also. But these are patterns. So the funny thing is we don't really know how we emptied how the mega herbivore niche was empty in Africa. We don't know if this is rising extinction, which is the default thinking, or is a combination of decreasing or low speciation. We don't know, because most of this discussion, both either if it's the humans were the main driver of the decline, or if it's climate the main driver of the decline, we all the people that, including us, talking about this thing, we have focused on higher extinction in larger things. So we basically have invoked a process or some mechanism that removes larger species, preferentially from the African herbivore species pool. But the truth is that when you empty, we can call it the mega herbivore niche or any adaptive niche, you can do this by several processes. You can do it by increasing extinction, or by decreasing speciation, or by a mixture of the two. Especially when you have a scenario of 4 million years of decline, where we know that many things can happen, and when you have a multi-million year process, you really need to look at the two facets, both extinction and speciation. And of course, so for this project, we wanted to look at a larger time span, so not just the last 5 million years, or 7 million years, which is what most of the people have done so far. So we're looking at everything from the Miocene, so the last 23 million years. So we are looking at changes in aridity across this time interval, and how this can impact speciation and extinction in body mass. But of course, we know that diet may be also something very important, because we have some knowledge that browsers of things that are restricted to eating, fruits and leaves, so more linked to forested habitats than to really more open or open ecosystems like savannas or something like that, they may have been disappearing faster. So we know that diet may have been a very important modulator to this. And also phylogeny, because we know that not all the lineages, not all the families, not all the lineages of African herbivores have the same inherent potential for speciation and extinction. So of course, whatever the interactions between body size and environmental forcing and diet, it may differ and change across the tree of herbivores. So, of course, this all sounds really good, but the truth is that we could not put everything together in the same diversification model. You had to pick one variable or the other, or the environmental variable or some trait, and put them in a different model. So we didn't have a way to really rank the importance of all these variables in a given model. But now we have this model that Thorsten Haufe developed, and also I'm glad to be part of this very useful paper where we were putting together a model based on neural network. This is a neural network expansion to Pyrate, where you can really fit all these traits, both species traits and time-dependent variables, and then combine all your data in the model. So this model opened the door to this project, because this is something that we could not have done before. So just a brief glance at the data. We are modeling around 400 species with 3,300 occurrences. We have body size in 6 categories for all these species. We have ipsodonty, which is the relative height of the molars, and this is proxy for dietary plasticity, because things that get more ipsodont, they really are able to cope with harsher or more abrasive food, so they are able to incorporate more grass to their diet, or even getting completely specialized in just eating grass, as many antelopes. And also they already mentioned proxy for aridity. over the Neogene and the Quaternary. And as I said, one of the cool things about this model, and I will be showing you results, but I'm really talking about the method, because this method allows you to do so many things that the only way for me to explain it to you is to show you plots. So the first thing is that we can rank things. So for speciation, we have a ranking of the importance of each factor. We can see there is a lot of shared importance for speciation, but for extinction, we have like changes in aridity are very important and in explaining how extinction changed in African herbivores across this time span. But one of the cool things about the pirate neural network also is the proper name, I think, is bird death neural network, so it's BDNN. The BDNN pirate is that you can then not just rank these things, but then isolate the effects. So you can think about this model as a huge regression where you can isolate the effect of each variable and what slope it has or whatever. So let's look at the changing environment. We can just isolate the effect of aridity, or which is here measured as rate of dust flux deposits in marine cores, and how these speciation and extinction rates change through time. So we see that speciation basically remains flat across all the values, but extinction undergoes a threefold increase as aridity increases. So this is why aridity is the major driver of extinction in this system. And then there is a point here which we estimate to be around in the beginning of the Pleistocene, where just this variable alone predicts a net diversity loss in the system. So in general, if we just look at the effect of this variable, we will start losing species across all African herbivores starting between 2.5 and 1.8 million years ago. And also we can look at the effect of size alone, independent of any other factor. And the funny thing we see here is that larger things, they speciate at a slower rate, but they go extinct at a slower rate too. So this means that there is any intrinsic higher extinction risk in these larger things as we see today. So this means that all the higher extinction risk that we see today in larger herbivores and larger fauna in general in the planet is not something we can just bring to the past. It's not an inherent feature of larger herbivores. It's something that depends on the context. Now, a cool thing that we can do is we can start looking at interactions. So this is, if you remember this curve where aridity just drove a threefold increase, we can look at this in different size categories. So what we see here is that smaller things, they start responding before to the increase in aridity, and they reach slightly larger extinction risk than larger things. So actually larger things, even in the moment of higher extinction driven by increasing aridity, they only show 2/3 of the extinction risk of the smaller things. So what this is telling us is that even with environmental forcing, larger things, they were not experiencing higher extinction risk than smaller things. So why do we lose so many things, so many larger size species in Africa in the last 4 million years? And the reason is because they have very low speciation, as we saw before, but also in some cases, we see that increasing aridity actually have this kind of suppression effect. It's not super dramatic, but we're talking maybe a suppression around 25% of the speciation potential that they have at the beginning when aridity was lower. And actually what we see at the end when aridity is really extreme is that larger things produce species at less than 0.5% of the rate that small species do. So basically, what happens now that you have these two plots is that when you have this very low and suppressed speciation, even if your extinction is not among the highest, it's much easier for extinction to surpass this speciation level and then drive that part of the herbivore niche to negative diversification rates. So the negative balance is reached before, even if extinction is more moderate in larger things. So you can see that one of the interesting is that the interaction between body size and aridity here is much interesting because you have a more diversity of trends compared to extinction. Extinction is pretty much the same for all the body sizes if you compare it to its effect on speciation. We can look at this ipsodont index, the type of molars that these herbivores have. So we see that again, extinction is kind of homogeneous. The trend, everybody goes extinct faster when aridity is higher. We have low crown molar species going faster a little bit more, but it's not super crazy. But for speciation, this is crazy because we see a major difference. So ipsodont things, they really take, especially until moderate levels, they really increase their speciation capacity as aridity reaches to this moderate level, and then they follow back again. They have this unimodal kind of distribution. So these are isolated, let's say, effects. So we've been looking at body size and aridity with body size and aridity with ipsodont. Now, the cool thing is that we have so much information in this model, we can really extract it as we want. So basically, the next thing we were thinking to do is, okay, we have 18 combinations of traits. We can call them ecomorphs. So we can actually extract rates through time, like, more in like the classical way. We can use the information that the model encapsulates to extract rates through time for each of these 18 ecomorphs. And this is something that, when Thorsten developed this, I was like, okay, this is just fantastic, because now we can just look at the speciation over time across the ecospace and see where speciation was faster or slower across time. I'm focusing here on the last 7 million years, which is where the trend really gets more interesting in terms of extinction. So you can see that, again, larger things, they speciate slower, and especially if you are in the left part, which is the brachydont part. So large brachydonts were speciating slower compared to the rest. Looking at extinction, things become much more interesting because we don't have a huge change in the top part, but definitely the extinction plots are very interesting. Especially, as I said, starting around 2.5 million years, the Pleistocene extinction skyrockets across the board. So it's pretty much very unselected. In the beginning, it was focused mainly on small brachydont things, so the lower left corner. But then after 2.5 million years, everything gets crazy. So you can see here very clearly that extinction was not very selected. This is why in the previous plots, all the curves were really kind of, very close. And of course, we can get the net diversification where you can see in reddish colors things that start losing species. And you can see that in the beginning was mainly brachydont, the leftmost column of each plot. Again, going in age, I forgot to say this, but in the top you have ages, so going from 7 million years to the recent, from left to right. And after 2 million years, 2.5 million years, basically we have extinction hitting hard in, here maybe it's the largest brachydonts, and this is probably just one species that goes extinct, like the Deinotherium. But the rest is kind of, there's not a huge signal for extinction and net negative diversification. Only after 0.7 we see really that because speciation was lower in the top part, extinction was still very homogeneous, you have certain up and down gradient in that part. But it's not because extinction only, it's because speciation is lower in the top part. And then again, we can, of course, because we have phylogeny, and I won't get into much detail, but we can still keep unfolding this. So we have here 12 herbivore families in Africa. Again, temporal bins in the top, and again, the same 6x3 eco space where we can really track what's going on in terms of per capita speciation and extinction rate. So the blue is speciation, the red extinction, and in the right part we have net diversification. And then we can see how each group basically behaves in terms of macroevolutionary rates. Things that are interesting here to see is that even in the top part, you look at proboscideans, which are classic examples of mega herbivores. Their extinction is systematically not larger than if you look at suids or wild boars, or if you look at horses or things like this. They are not showing extraordinary high extinction. The highest extinction is actually recovered in things like wild boars and hippos and giraffes and things like this. But it's because they have really slow speciation. They have this pale color, this pale blue color in speciation. And the same goes for calicotheres, which is, you can see there is a perissodactyl group between horses and rhinos. They all go extinct because they also have this really low speciation potential. So this model with all this information on the very fine record of the African localities, they help us to really go to this fine grain and look at what's going on in each ecomorph, essentially across all these phylogeny. So just to wrap up, the main conclusions of this last part is that for the first time, we're looking at not just the effect of extinction or the drivers of extinction, but also the speciation. And we find that this is critical because we see that the speciation was critical to explain the decline of mega herbivores. That environmental forcing was a major factor shaping the faunal turnover in Africa, including this emptying of the mega herbivore niche. That the expectations of so many papers, including papers of mine, are not met, is that these things are not going extinct faster. They are just having a different way to behave in terms of speciation as environmental forcing ramps up. And that we have a lot of interesting interactions based on phylogeny and the different ecology, so that ecology and phylogeny are very important factors to understand this faunal turnover. And that this polyphacetic kind of scenario makes very complex, I think, to map the contribution of humans inside all these longer term trend of mega herbivore decline. I think it's gonna take a lot of data, or I don't know, like ecological modeling, even based on abundances and things like this, to really try to tear apart the role of humans in the extinction of some populations or some species. And yeah, with this, I thank you all for your time and for listening, and I'm happy to take any question.